Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Moira Lynch from Thermo Fisher Scientific and George Tan from Bristol Myers Squibb.
Thank you, Leah, for that introduction, and welcome to everybody who is in the audience listening. My name is Moira Lynch, and I'm an innovation leader for the bioproduction group within Thermo Fisher Scientific. We offer solutions across the entire bioproduction workflow, but today we're focusing on purification solutions for downstream processing. I sit within research and development, and I've been a part of strong technical teams that are developing new resins. In recent years, my teammates and I have launched capture chromatography products such as Poros AVX and Poros Oligo DT25, and polished chromatography products like our family of Poros HIC resins. Today, we will share collaboration work with one of our anion exchange resins, Poros HQ. But before we get into that, I'd like to take a moment to set the stage for the body of work that we will present today. Viral clearance studies are an important required activity during the development of biotherapeutics. These studies are typically happen at two critical stages of development. The first is just prior to phase 1 clinical trials with just a handful of model viruses. And the second is just prior to phase 3 clinical trials with a wider selection of model viruses. Viral clearance studies are designed with specific goals. They must include a selection of relevant viruses. If you're working with a CHO system, you need to be working with viral clearance studies that include relevant viruses for a CHO system. The studies will typically be performed under the worst case scenario to really challenge the system. And keep in mind, the entire downstream process needs to demonstrate at least 10 to 15 logs of reduction of virus. So that means each step of that process needs to show at least 2 to 5 logs of reduction. These studies are conducted at specialized third-party facilities that have equipment and expertise to handle live viruses. And because of the specialized nature of this work, the studies are understandably expensive. But for developers, that high cost tends to push these studies out until they are absolutely needed. Now let's look a couple steps back and look at process development from a viral clearance perspective. For sufficient viral clearance, it's recommended to include at least 2 orthogonal modes of separation. Most process development teams will base their choice of separation steps on viral clearance that has been reported in the literature. And then prior to phase 1 clinical trials, process development must have a representative scaled-down model of their final process so the scaled-down model can be used in viral clearance studies. But the process development group will have no way of knowing how their final separation step actually performs for removing viruses until much later when that first viral clearance study is performed. Anion exchange chromatography is a well-established polish step for viral clearance and removal of other impurities. For target molecules that tend to be basic, such as monoclonal antibodies, anion exchange makes perfect sense to be included in the process. This polish step can be run in flow-through mode such that the pH of the loading buffer is optimized so that the target molecules flow through the column and acidic impurities such as DNA and viruses bind to the column. At Thermo Fisher Scientific, we offer 2 strong anion exchange resins suited for this purpose, Poros XQ, which is a fully quaternized amine surface chemistry, and Poros HQ, which has 60% quaternized amines with the remainder surface chemistry made up of a blend of other amines. This blended amine surface gives Poros HQ a unique selectivity. Like most resin vendors, Thermo Fisher Scientific performs viral clearance studies using samples and buffer conditions that are representative of the way our customers will use our resins. Our customers will have to perform their own viral clearance studies when the time comes, but offering data like these help our customers understand what type of viral clearance they might expect if they use our resins. The top table shows data from a viral clearance study we did for Porous XQ and Porous HQ. Protein load, flow rate, and column height were kept constant. And we looked at how increased load conductivity affected the viral clearance of 2 model viruses. You can see that for XMULV, decent viral clearance was achieved at all conductivities for both resins. However, MMB showed some sensitivity to conductivity. The bottom table is Porous HQ viral clearance data kindly provided by a customer. In this study, a shorter column was used at higher flow rates. Under these conditions, decent viral clearance was consistently demonstrated for 3 model viruses used in the study. But enough background, let's get to the good stuff. At Thermo Fisher Scientific, we have a culture of collaboration. I have personally experienced the value of customer collaborations during product development. Customers who are willing to test some of our earlier prototypes and give us honest feedback, helping us make sure our development is heading in the right direction. A couple years ago, we had a great opportunity to collaborate with Bristol Myers Squibb. George and his team approached us for an interesting proposition. Would we like to collaborate with them to map the viral clearance operating space of Porous HQ? How could we say no? And with that, it is my pleasure to introduce George Tan from Bristol Myers Squibb to share more about our collaboration. Thank you, George.
Yeah, thank you, Moira. And that's a very good introduction in terms of Porous HQ resin and how we initiated this kind of collaboration. So my name is George Tan. I work for Bristol Myers Squibb at Demis. And my focus is really to optimize the process downstream development. So now I want to give you a little bit of background regarding this collaboration study between Bristol Myers and Thermo Fisher. So as you all know, this viral clearance is really necessary for the IND and the BLA filings. And the viral clearance often being done in the CROs after the clinical manufacturing. And it's very expensive, we all know. And but more importantly, sometimes we get the better results after the viral clearance, but by the time it's too late. So in order to really make sure those viral clearance results deliver as we expected, we want to make sure the AEX process is very robust. As you can see on the right side, this is a typical downstream process flow. AEX as a workhorse, not just to remove the typical process impurities such as host cell protein and DNA. It's also very robust, normally very robust viral clearance step, just like a viral filtration. So in order to really achieve a robust AEX viral clearance process, at that time, we were developing the downstream platform. So we want to make sure the resin we select is able to give us a consistent, robust process. And so we reached the Thermo Fisher and really focused on these objectives for this collaboration. First, we focus on the AEX, the chromatography step. Secondly, through this study, we want to gain the good understanding of the viral removal mechanism, and the ultimate goal to enhance the viral removal capability and deliver the process robustness. Before I dive into the more data, I wanted to deliver a few key takeaway messages from this study. First, we used a DOE approach to conduct a viral clearance study and to further understand the viral removal mechanism on the AEX chromatograph. Secondly, through this study, we find out that just based on the molecule PI to decide the operating pH and the conductivity may not be the best way to achieve high viral removal performance. Third, so we propose the virus co-elution mechanism based on the mAb-virus interaction. This mechanism can help to explain some unique observations encountered in this study. Lastly, our virus co-elution mechanism is able to explain interactions among resins, virus, and the protein. This additional understanding will improve the viral clearance capabilities for broader molecule classes. So for this viral clearance study, we intended to achieve maximum value with limited number of experiments. As we all know, it's very expensive. So therefore, the experimental design is very essential. For this purpose, we took a DOE approach, trying to use molecules with different biophysical properties. And at the meantime, we choose MULV and MMV as two model viruses. But our focus is really on the MVM, which is considered as more challenging virus for the downstream removal. As you can see on the table on the left, we list the 3 molecules we selected for this study, and with a wider range of the PI, and also we also include the hydrophobicity as one of the biophysical property for these molecules. And for the better illustration, so I put this kind of figure on the right side to give you a better idea to where these molecules are in comparison to the virus. So overall, for the maps we selected for this study have relatively high PI compared to the virus. But on the purpose, we selected 2 high PI molecules, the B and C. But the B and C has different hydrophobicity. So hopefully, through this kind of design, we are able to not just like look at the AEX in general, but we are able to really draw some kind of correlation between the molecular biophysical properties and the overall viral clearance capability. So once we have a big picture regarding our goal, and here come to the detailed experimental plan. So you can see here, Thermo Fisher provided the porous HQ resin, and they are very nice to provide the pre-packed column, which here we use the 0.46 centimeter diameter and the 20 centimeter column height for all the studies. And the Bristol Myers, we provided the materials, like the 3 maps with different PI and the hydrophobicity. And also for all the buffers, we made it and we covered the pH with a range of 7.2 to 8.2. And also the conductivity, the range of 2 to 8 millisiemens. As you can see, our primary goal is really for the MVM, but we also add a little bit experiment for the MULV. So with 10 rounds for the MULV, and with the use 1 molecule at the 3 factors, use 3 factors and at the 3 levels. For MVM, we did 24 rounds, and with 4 factors and 3 levels. For all the studies here, we normalize the protein concentration to around 8 grams per liter. In a sample loading from the 200 to 500 grams per liter resin, for all the rounds, we have same resin time at 4 minutes. The table on the right side just shows all the rounds we conducted for this study. So this shows the DOE results from the MVM experiment. Overall, a pretty good fit between the experimental viral clearance ARVs and the predicted ARVs. As you can see, the model fitting achieved R square greater than 0.8. It's pretty good fit. And if we evaluate the study altogether, you can see the most significant factors are molecule properties, compounding factor of pH and the molecule, conductivity followed by pH. Later on, we will decouple those factors per molecule, and which will show each factor may have different influence on the overall bioclearance ARV. So here we break down into each molecule and look into the bioremoval ARV response against the pH and the conductivity. As you can see, the low PI map, as you can on the left side, and it has a wide range of the ARVs from the 2.2 to 4.6. Specifically, the ARV was sensitive to the pH and the conductivity. You can see a pretty big slope. And with the increase of the pH and the conductivity, the ARV for map A increased significantly. On the other hand, for map B, which is a high PI molecule, but with a low hydrophobicity, and we achieved a range of the ARV 2.7 to 4.6, but most of data are really 3.6 to 3.8. As you can see, the ARV is insensitive to the operating pH, but slightly increased as the operating conductivity increased. Interestingly, for map C, which you can see on the right side of this screen, this is a high PI molecule, but also with a high hydrophobicity. We achieved the overall high ARV. But I just want to mention that for this molecule, we achieved the overwhelming virus counting in the flow-through below the quantitation limit. As a result, the ARV calculation, most of the data points show that greater than certain numbers. As you can see, just from those plots, the ARVs were independent of the operating pH and conductivity. In other words, those ARVs are insensitive to the operating conditions. Clearly, through these three molecules, they show the different response to the operating conditions, which may suggest molecule biophysical property might influence the outcome. If this assumption holds true, specifically, what kind of property influence the ARVs and why? So before we went to a deep dive into the speculation of why different molecules behave differently, or why similar maps like your map B and the map C, they show different levels of ARVs. And let's first revisit a traditional AEX chromatography, how that works. So for a typical AEX, so you have the maps normally have the high PI, or even have the low PI maps. The overall PI is still higher than the virus PI. And for the typical operation condition, you have a pH 7 to 8, that kind of range, and you would have the high PI molecule have the positive charges, and the low PI molecule may have the negative charges. So because of the nature of this, and for this typical AEX operation, you would have a positive charged AEX resin, and also coupled with a negative charged virus bind on the AEX resin. And for the most of the maps with high PI, those positive charged maps will flow through the AEX resin. At the meantime, these maps have this a positive and net charge will cause greater repulsion between the map and the resin. And this will promote the map to flow through more easily, and then enhance the map and the virus separation and lead to the high ARVs. However, for those molecules which have the lower PI, and in this case, the pH, operation pH will be above the MAP PI, and the MAPs will have a negative net charge. And this will promote the MAP resin interaction and a possible MAP virus competition for the resin binding sites. This would lead to the lower ARVs. So the high PI molecule and the lower PI molecule would have different outcome under this typical AEX operation conditions. So now let's compare the traditional observation from the AEX resin and also the current AEX DOE results. The first for the MAP-A, which is a low PI molecule, but it's a hydrophobic molecule. We do expect to see the low clearance ARV, and the actual results did show it's a low ARV. But one thing we didn't expect, when the pH increased, also the connectivity increased, the viral clearance ARV increased dramatically from 2.2 to close to 5. That's very significant. For the MAP-B, it's a high PI molecule. We expect the high ARV, but we only expect the moderate ARV from the actual experiments. And the one thing like we observe, when the pH and the connectivity increase, and the ARV remains largely unchanged. And for MAP-C, we expect a high ARV, and our experiments also demonstrate its overall high ARV. One thing, like when the pH and the connectivity increase, and it's similar to the MAP-D, the ARV remain largely unchanged. But how this discrepancy between the expectation and actual results can be explained? So there are a few questions that we really ask ourselves. First, how can the high ARV be achieved for the low PI molecule? In this case, MAP-A. So understanding the mechanism can potentially provide an optimal operating space in the future to improve viral clearance for the challenging low PI molecules. Secondly, for the high PI MAPs, such as MAP-B and MAP-C, the operating below the PI of both MAPs, although we are able to obtain generally high ARV, why there's a large discrepancy between these two molecules. Lastly, the conventional expectation and understanding of the AEX may not be able to explain the phenomena observed here. In addition to the traditional resin virus and the resin MAP interactions, other mechanisms may exist to help drive this kind of a unique phenomena here we observed here. So now let's take a look at the molecule of properties. So in BMS, so we have a biophysics group specializing in the molecule modeling, which can help the process development and also the analytical development to understand the sort of observe the behaviors at the molecule level, which really give us a big help. So first, from this slide, you can see we have the molecular surface charge profiles for the three maps at a different pH. On the top, that's the molecular surface charge profile at the pH 7. And the bottom, so we have the same surface charge profile at the pH 8.5. So firstly, you would see the three maps clearly show different positive and negative charge distribution when they are exposed to pH 7 and the pH 8.5. So for example, at the pH 7, MAP-A showed a very large positive charge patch, as it shows in the red circles. And then for the MAP-B and the MAP-C, in addition to the red circle on the left here, there are other positive spots. Secondly, when you change the pH from 7.0 to 8.5, you clearly see some positive charge patches disappeared. Besides the surface charge distribution, this is the same biophysical group was able to do the molecule modeling to determine the hydrophobicity patches on the protein surface. The hydrophobic patch is highlighted as like red, and also the hydrophilic patch is highlighted as blue. As you can see, MAP-A and MAP-C have significant hydrophobic patches in the CDR region, which is in agreement with our experimental results determined by KIC HPLC that MAP-A and MAP-C are more hydrophobic molecules. So here, in order to link the charge distribution with the hydrophobicity mapping, and here I so-called those previously positive charged spots. As you can see, there is additional positive patches that's different between the molecule B and C. And what does this tell us? This tells us that some of the positive patches are located in the hydrophobic regions and some in the hydrophilic regions. And because of the distribution difference, this may lead to the overall bioclearance results difference. With that prelude, that the molecule surface biophysical properties may influence the overall bioclearance ARV, we are adding one more dimension of the interactions among resin, MAP, and the virus during the AEX flow-through chromatography. As you can see on the left, traditionally we often discuss the MAP and the virus separation by only considering the virus resin interaction and the MAP resin interaction. As such, we modulate the process condition to enable the maximum virus resin binding and minimize the MAP resin interaction. There is nothing wrong with that kind of approach. However, this just cannot explain the observation that MAP-A, which is a low PI molecule, and that can achieve high ARV, and also the ARV increases as the pH and the conductivity increase. Not mention that the MAP-B and the MAP-C show the distinct ARVs given their very similar PI values. Therefore, here we propose the following mechanism. So first, as you can see on the right side, there is a potential attractive electrostatic interactions between the MAP and the virus. This can influence the MAP virus separation, hence to influence the final overall viral removal. Secondly, the MAP virus interaction will not contradict to the classic ion exchange effect that lead to the retention of the virus on the resin and the flow-through of the MAP. Quite opposite, the MAP virus interaction mechanism would improve the overall AEX separation understanding by taking consideration of biophysical property of the molecule and what kind of influence they may have on the bioclearance. With that, this will help to enhance the overall bioclearance ARV and improve the overall process robustness. So with that, we propose this co-elution mechanism, meaning the virus and the MAP may flow through the AEX column together. So the co-elution is induced by the MAP virus interaction due to the specific biophysical property of the MAP. As you can see on this figure, under the AEX flow-through mode, the resin is positively charged, virus is negatively charged. The MAP molecule would have both negative and positive charged patches on the surface. As we all understand, those positive and negative charged patches are influenced by the environment. However, due to the unique biophysical property of the molecule, the charged patch distribution may be impacted by the biophysical properties, which also is influenced by the surrounding pH and the conductivities. A transition state, that's the virus and mAb interaction caused by the protons and also ions shielding, may become pronounced. As a result, the virus flows through the column with the mAb molecules. So now, how this coelution mechanism can help us to explain the behaviors of the 3 mAbs on the AEX resin we observed. So let's start with the first molecule, molecule A, which is a low pI and a hydrophobic molecule. As you can see, that when the pH changed from 7.2 to 8.2, we saw the VC ARV drastically improved from 2.2 to 4.6. Why did this happen for this low pI molecule? With that kind of question in our mind, we then started to pay attention on the protein surface charge, and that kind of modeling to give us some kind of idea. We noticed that a cluster of positive charge in the CDR region at pH 7 disappeared when the pH changed to 8.5. If we look at the CDR region as a whole, you will notice that that's the only change. At the pH 7, large positive patch may promote attractive mAb virus interactions, which may increase the coelution effect. That's why this may result in a lower ARV. On the other hand, when the pH increase from 7.0 to 8.5, such a virus mAb interaction are eliminated due to the neutralization of the positive patch. Therefore, change of the virus coelution effect was diminished. Therefore, the overall ARV increased as we increase the pH and the conductivity. So it appears that the virus coelution effect can explain the low pI molecule. How this is going to explain for the other 2 molecules which have the high pI. Now let's look at the MAP-B first, which is a high pI but low hydrophobic molecule. As you can see, we changed the pH from 7.2 to 8.2. The moderate ARV was achieved. And then when you change those conditions, the ARV value remained largely the same, unchanged. So we use the same approach when we did for the MAP-A. By looking at the surface charge map, in the study condition, we noticed that negative charge patches showed no change, and a very small change on the positive charge from 7.0 to 8.5. The very little change of those positive and negative charges would suggest a very little change on the mAb resin interactions or mAb virus interactions. As a result, the viral clearance ARV showed relatively insensitive to the pH and the conductivity changes. But how can we explain the overall moderate viral clearance ARV for the MAP-B? With a good understanding of the molecule is less hydrophobic. The majority of positive patches remain unchanged. Then the weak virus mAb interaction would lead to the overall weak virus coelution effect, which may explain the moderate ARV for the MAP-B. So finally, let's take a look at the MAP-C, which is a high pI and more hydrophobic molecule. So when we change the pH from 7.2 to 8.2, we observe the overall high viral removal capability, and the ARV remained high across the entire study range. However, compared to the MAP-B, significant high ARVs were achieved. Despite these 2 molecules have very similar pIs and charge distribution. But when we look at the MAP-C, all of the positive patches in the CDR region are located in the neutral or moderate hydrophobic area, in which the interaction between the charged sites and the virus become unfavorable. But for the MAP-B scenario, all the positive patches are located at the hydrophobic region, in which the interactions between the charge site and the virus are more favorable. In that case, this will promote the virus coelution effect. Keep in mind that for the MV virus, they are in general hydrophilic. The overall virus MAP interactions will be subdued for the MAP-C molecule, which is a hydrophobic molecule. This will result in the overall weaker virus coelution effect. This will give us a consistent high virus clearance ARV for MAP-C. Through this kind of discussion, it seems that using this virus coelution mechanism based on the weak virus MAP interactions is able to explain the behaviors we observed for the 3 maps used in this study. That sounds great. However, oftentimes we want to develop a robust process to achieve good impurity removal, including not just the virus, but also the host cell proteins and also the residual DNA. One argument we often have is that the virus and the HCP DNA may compete to bind on the AEX column, which may give us the challenge. Like sometimes we get the poor viral clearance results, we blame the HCP and DNA. And for the same reason, people would suggest that the impurity clearance profile we are able to achieve in our lab, this may give us some prediction about the viral clearance, if you really consider this competition theory is true. Because of this, we did exactly the same AEX round in our PD lab and tested the HCP levels and plotted the HCP versus the pH and the connectivity, as you can see on the slide here. Clearly, HCP clearance does not correlate well with the viral clearance results. But one thing I want to mention is we observed in this study, we observed the impurity levels in our loading material for the HCP and DNA are very low. So which may not be the case like for the competitive reason. And those HCP and the DNA are so low that this competitive binding against the virus is very hard to quantitate. So I guess just through the HCP and the DNA determination, we cannot predict the viral clearance. We still have to do the viral clearance study. So here I come to the conclusion of this collaborative study between the BMS and the Thermo Fisher. First, we use a DOE approach to study the viral clearance on the AEX chromatography. This study provides us very useful information to gain a good understanding to achieve a high viral clearance ARV. Secondly, although it is AEX chromatography, the conventional factors solely rely on the molecule PI and adjusting the pH and the conductivity may not achieve optimal viral clearance results. For that same reason, we propose a virus coelution mechanism based on MAP virus interactions. This can help us to explain the strong viral clearance predictions for the 3 maps used in this study. Fourth, the molecular properties, namely surface charge and hydrophobicity, in combining with the operating pH and the conductivity, all those combined together can influence the MAP virus interactions. With this additional mechanism, with previously virus resin interaction and the MAP resin interaction, all these 3 interactions contribute to the overall viral clearance performance. Again, lastly, take the process development altogether. We need to achieve a good clearance of all targeted impurities, including viruses, HCPs, DNAs. We need to reach a balance of good HCP and DNA clearance, which can be determined in our PD lab. But the viral clearance ARVs need to be done in the CROs. So therefore, this good understanding of the factors and their impact on the targeted impurities removal is very necessary to improve the process performance and the robustness. So moving forward, we think we can still focus on other areas. Primarily, these 3 areas we identified: the enhanced process performance and the robustness, to develop the generic bioclearance strategy and develop alternative bioclearance surrogates. Specifically, to enhance the process performance and robustness, we can adjust the AES operation conditions to reduce the potential MAB virus infection. And also, we should be able to achieve a high bioclearance ARV for the challenging low PI molecules. For the generic bioclearance strategy, in the context of the MAB virus interaction mechanism, generic class of bioclearance behavior as a function of the molecule characteristics, namely surface charge and hydrophobicity distribution, may become evident. This improves the mechanism here, really help us to understand some additional AES bioclearance results for a wide range of the molecules. This kind of strategy may, in the future, can be applied for the modular, use this kind of modular approach for the regulatory filing of the bioclearance strategy. For alternative bioclearance circuits, I use the mock of virus particles as the alternative surrogates for the broader bioclearance study, may provide a deeper insight of the coelution mechanism. Last but not least, I want to thank to many people who have made great contributions to this study and made this collaboration successful, including my colleagues in BMS and my friends at Thermo Fisher. Thank you all.
Thank you, George. That's excellent. It's been a pleasure collaborating with you, and it's been a pleasure to share our collaboration with the audience today. And with that, let's move on to questions and answers.
Okay, great. So the first question is, how do you decide on the design factors you include in your design of experiments?
Yeah, I think that's a good question. So as we all know, operating pH and the connectivity would impact the process performance, including impurity removal, such as HCP DNA, as well as like a virus removal. However, when we perform the bioclearance validation in the CROs, we often just have a molecule specifically like defined range for the operation. So different molecules, you may have different operation conditions. Due to that, it's not really practical to compare the ARV results across different molecules. So with that in mind, we wanted to have a uniform way to really look into whether molecule may play some role. So we needed to control the process conditions. And we figure out the DOE approach will be the best, a most effective way to investigate if any correlation exists among those factors.
Okay. And you indicated moving towards filing generic bioclearance packages. Can you expand on that and share how that might be put into practice?
Yeah, sure. I think I very briefly touch upon some rationale in our DOE design. And we strategically chose molecules including low PI MAP and the most abundant high PI MAPs to try to cover the broad spectrum of molecular properties. The main reason of such decision was to evaluate the molecule property impact on the viral removal ARV under a specific operation space. In our case, we investigated the ARVs under the pH ranging 7.2 to 8.2, and the connectivity 2 to 8 millisiemens across 3 maps with different molecular properties. With the virus coelution mechanism, as we proposed in this study, we were able to explain the behaviors for all 3 maps. And equally important, through this study, we explore operating windows that provide good viral removal ARV for similarly challenging low PI molecules. With a clear distinction of correlation of the viral clearance ARV and the molecule biophysical property under the respective operating conditions, this approach can potentially be used for generic viral clearance package if the molecules have a very similar biophysical property.
Okay. And have you changed the way you approach process development since this study?
Yeah, I mean that's kind of a whole purpose, So our original idea was we want to do a good homework before we start the lab work. So in that case, we strategically choose those molecules and to gain a good understanding. Now, this clearly demonstrates that kind of initial homework was paid off. So this is a kind of approach, we intended to implement for all the future molecules to do a thorough, like investigation, or not really investigation, understanding of the molecules, rely on molecule modeling to gain a further understanding at the molecule level. And this really can give us very high confidence, when we actually perform the, process development, and including the robust, viral clearance, results. So that's definitely will change our mind, So.
Okay. And after the study, do you think you could use molecule mapping or molecular mapping to help you predict viral clearance?
Yeah, I think so. At least, through these 3 molecules, we have pretty good idea, what kind of ARV we may achieve. And again, this is just based on 3 molecules. Our goal is to use more molecules with respective biophysical properties, and to further validate, those hypotheses. Hopefully, with n greater than 1, let's say 2 or 3 molecules for each category, and we are able to demonstrate that's the case. This is really not just give us high confidence, and potentially to be used for the the generic viral clearance fighting.
Okay. And how do you know which anti-exchange resin to use, porous XQ or porous HQ?
Well, it depends. There's a lot of evidence in the literature and even the results of this study that show that the target molecule potentially has a huge influence on the impurity profile. And so I think it's best to screen both resins and optimize for impurity removal for your particular target molecule, because each resin may offer a different selectivity and be better for your target molecule.
If someone has an idea for a collaboration, who do they contact at Thermo?
Oh, that's a great question. So if you have any ideas for collaboration, please reach out to your local sales representative or your local field application specialist.
Okay. Were you able to run all design of experiments in one day, or did you have to block across days?
Yeah, I mean, we cannot run all 24 in one day, and I think I remember correctly, we did molecule by molecule on multiple days.
Okay. And this one is for George. Does George know how the PI of each virus was determined in the studies for which he provided a reference?
Yeah, I mean, PI is determined by theoretical calculation based on the amino sequence, and the hydrophobicity is like based on also determination by the SAP score. So we have this kind of standard what should I say? The team to really determine the surface charge and also the surface hydrophobicity. So that's we have a software in place. So that's how we determine.
Okay. Would you suggest adding additives to break the viral MAB interactions in order to improve the viral clearance if changing conductivity and buffer pH do not improve viral clearance in some cases?
Yeah, I think I would consider that would be the final, solution. Consider the AEX step, normally either first polishing step or last polishing step. With that kind of addition of the additive, that may add some, challenges regarding the safety. So that's something you may have to address, from a safety perspective.
If the charge of the biotherapeutic protein is similar to the viruses, then what type of resin should we use for virus clearance?
That maybe Thermo has to develop a new resin.
I mean, I think that's the recommendation is to use multiple modes or, orthogonal modes of separation. You're not going to be able to disrupt all of the interactions with one mode of chromatography. So it will be based on your target molecule and the impurity profile that you're trying to reduce. And so, polish steps include cation exchange as well as hydrophobic interaction chromatography. Those can all be explored.
Okay. Did you model yield for the MAB products?
Yes, we have the results for the yield, and due to the relevance to this particular topic, we didn't put the data here, and we do have the results for the yield.
Okay. Well, it looks like we have time for just one more question. If we didn't get to your question, we will be passing those on to George and Moira, and they can follow up with you directly. So if you still have questions, go ahead and enter those. And the last question, these are all these great questions. So design of experiments was really important for you to gain AX viral clearance insights. What was the biggest challenge for you to implement design of experiments, and how do you think others could benefit from DOE use? And would you recommend it over conventional methods?
Yeah, I mean, for the DOE, this is really try to map out, some kind of a broad scope. If you don't really have a good understanding of the molecule, and the DOE is kind of a universal approach, and something you don't have the good understanding at the very beginning. However, if you already know the general trend, then you don't really have to use this kind of four-factor DOE. You can just run the OFAC, and based on the molecule property, for example, the high PI molecule, and in that case, you don't really have to try some conditions, it's not going to work. So this is DOE is just for initial screening, and for some unknown molecule properties may play some role. And with a good molecular understanding, you may not need this kind of DOE approach.
Okay. Well, thank you, George and Moira.
Thanks, everybody.
Thank you.
Thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye.
Bye.